1 minute, madam.
Yes, sir. Yes.
So can you see my slide, madam?
Yes, sir. Yes, sir. Visible, sir.
Okay.
Okay. Thank you. Shanmugapriya, ready? At 10:30 sharply you can start.
Sure, ma'am. Shall I start, ma'am? Ma'am?
Yes, ma'am. Check whether principal or vice principal is there.
Sure, ma'am. A very beautiful, pleasant, wonderful good morning to everyone present here. Blessing, a beautiful step in the journey of our destiny. So let's make our start with a prayer song.
सत्यव्रतं सत्यपरं त्रिसत्यं सत्यस्य योनिं निहितं च सत्ये सत्यस्य सत्यं हृतसत्यनेत्रं सत्यात्मकं त्वां शरणं प्रपन्नाः वाणी गुणानुकतने श्रवणौ कथायां हस्तौ च कर्मसु मनस्तव पादयोनः स्मृत्यां शिरस्तव निवास जगत्प्रणामे दृष्टि सदा दर्शनेषु भवत्तनूना नमो भगवते तस्मै कृष्णायाद्भुतकर्मणे रूपनाम विभेदेन जगत्क्रीडति योऽयम्
Thank you for this happier start. Now, I would like to welcome Ms. Ishika Sri to present the welcome address.
Good morning, one and all present here. It is my immense pleasure to welcome you all for today's webinar. On behalf of our institution, I welcome our chief guest, Dr. Busi Siddhartha, Assistant Professor, Department of Microbiology, School of Life Sciences, Pondicherry University, to present this webinar on application of nanoscale particles with photosensitizers for antimicrobial photodynamic therapy. Thank you so much, sir, for accepting our invitation and coming over for the webinar with your busy schedule. A special welcome to you, sir. I feel happy to welcome our principal, Dr. R. Geeta ma'am, Vice Principal, Self-Finance Team, Dr. S. Sita Lakshmi ma'am, Head of PG Department of Chemistry, Dr. S. Sugna ma'am, and faculty members of PG Department of Chemistry. I also welcome professors and assistant professors of other departments, and also Mansia ma'am and Padma Priya ma'am. Last but not the least, I welcome you, my dear students. And provides an idea to deal with the problem.
Thank you, Ishika, for your hearty welcome. Now, this is the time for us to explore the life journey of our chief guest, Dr. Bhuvi Siddhartha sir. I would like to invite Ms. Ramya Sri to deliver it.
A very good morning to everyone. I am more privileged and proud to introduce the chief guest of today's webinar, Dr. Bushi Siddhartha, Assistant Professor in the Department of Microbiology in School of Life Sciences, Pondicherry University. He completed his post-graduation from Andhra University, Visakhapatnam, and completed PhD in Indian Institute of Chemical Technology, Hyderabad. His areas of interest in research is bacterial quorum sensing, drug discovery, microbial biocatalysis, and microbial metabolomics. He was awarded Junior Research Fellowship, JRF, and qualified CSIR NET lectureship also. In the year 2002, he was qualified in Graduate Aptitude in Engineering GATE conducted by Indian Institute of Technology, IIT. He started his career
Ramya. Ramya. Ramya, what happened?
Yeah, might be some network issues.
Yes, sir. Yes, sir. Shanmupiya?
Okay.
Yes, sir.
Okay.
in the year 2002. He was
Thank you, Ramya, for sharing this mind-blowing journey. Now, I'm very glad to welcome Dr. Busi Siddhartha sir to carry over the session.
Sir, please, sir.
Thank you, madam. Thank you for a nice introduction. Okay, I'll start my presentation.
Yes, sir, please.
Is it visible, madam, my slides? Do the slides are visible?
Yes, sir.
Okay, madam. Thank you. So I'm Dr. Busi Siddhartha, working as assistant professor in the Department of Microbiology, Pondicherry University. My areas of expertise are microbial biofilms, drug discovery, quorum sensing, and nanotechnology that is related to the photodynamic therapy. So today I'm going to deliver a talk that is related to the application of nanoscale particles with photosensitizers for antimicrobial photodynamic therapy. Even though I'm going to talk something about biology application, okay, it is more related to the chemistry or material science, where the interface of nanotechnology and a photosensitizer, that is a dye combination, how we can able to employ them in order to kill the bacteria, in order to use as in medicine, and in order to application towards the antimicrobial therapies. So the entire talk is divided into two categories. One is about multi-walled carbon nanotubes, and second one is application of Mesoporous silica nanoparticle. So we used these two flags, either multi-walled carbon nanotubes or Mesoporous silica nanoparticles in combination with photosensitizers, and we studied their efficacy against bacteria like Staphylococcus, Pseudomonas, and even fungi Candida albicans. So my first discussion is about application of antimicrobial photodynamic therapy and Pseudomonas aeruginosa and Staphylococcus aureus using photosensitizer doped carboxy-functionalized multi-walled carbon nanotubes. So we doped some of the photosensitizers with multi-walled carbon nanotubes. Okay, you know that carbon nanotubes are gaining more importance nowadays in medicine. We explored these multi-walled carbon nanotubes along with photosensitizer to kill two highly priority pathogenic organisms, they're Pseudomonas aeruginosa and Staphylococcus aureus. So coming to the introduction, what is a bacteria? What is the problems with the antibiotic resistance, and how some alternative technologies are involved? So the global challenge of pathogens such as influenza, tuberculosis, and emerging human immunodeficiency virus and infectious diseases remains most important problem. Now we are facing the coronavirus. So therefore, antibiotics are extremely valuable resources across the whole spectrum of modern medicine. New resistant mechanism is acquired to the existing antibiotics like tetracycline, penicillin, carbapenem, and etc. So because of this multi-drug resistant, the disease is prolonged, disabilities and deaths are becoming more. So multi-drug resistance is because of various patterns like quorum sensing or because of biofilm formation, which makes these microorganisms resistant to the existing drugs. So these infections are associated with an approximately twofold higher death rate and considerably prolonged hospital stays. So in order to prevent this, as antibiotics are not efficient, we need some alternative. So photodynamic therapy is gaining importance. Photodynamic therapy is already in clinical trials in cancer therapy, but here we used antimicrobial photodynamic therapy. That means we are using photodynamic therapy towards bacterial infections. So even nowadays, you can see a lot of goggling solutions in the dental hospitals are related to the photodynamic therapy. They are in the clinical trials. So antimicrobial photodynamic therapy is a very promising method to combat bacterial infections, even for resistant strains. So I can call antimicrobial photodynamic therapy as APDT. APDT involves use of harmless visible light, with a particular wavelength, and a light sensitive dye that is excited because of the particular wavelength of light, that is called as a photosensitizer, and oxygen present in and around the cell, that is within the cell environment or outside the cell environment. This makes to prevent the bacterial infection. Fortunately, no resistance towards photodynamic therapy was observed because it is not working on a particular mechanism. It is having a multitude directions. It can able to stop the DNA, or it will degrade the DNA, protein inhibition, or cell lysis. It has been proposed that ROS, that is reactive oxygen species, generated by light-activated photosensitizer, can trigger microbial killing and cell damage via three mechanisms. Either they will damage the cell membrane, or they will inactivate the essential enzymes and proteins, or they will damage the DNA. Because photodynamic therapy, ROS generation, is working on multitude targets. Okay, it is a very promising, and no resistance can be observed against this photodynamic therapy. So this is the schematic representation of photodynamic therapy. Okay, we classify photodynamic therapy into type 1 and type 2 photodynamic therapy. In type 1 photodynamic therapy, okay, charge is transferred to a substrate or a molecular oxygen is generated, reactive oxygen species, like hydrogen peroxide and oxygen radicals, like superoxide ions or free hydroxyl radicals. Whereas in type 2 photodynamic therapy, energy is transferred directly to the molecular oxygen, whereby highly reactive singlet oxygens will be generated. Means we'll take a photosensitizer, we'll expose to a particular wavelength of light, they will go to the higher level, and because of this, they will produce the ROS. This ROS, the reactive oxygen species, will work on different macromolecules of the cell to kill. So this is the main concept of photodynamic therapy. So we used nano platforms like multi-walled carbon nanotubes. You know that it's a sheet of nanotubes one by one on top of other that makes a multi-walled. They have an inherent property of antimicrobial property, as well as they are good in producing the ROS. So we want to combine multi-walled carbon nanotubes along with photosensitizer to kill the bacteria. So that is the main objective of our study. So how this photosensitizer delivery and nanomedicine works. So one of the most important problem in PDT is the solubility or the delivery of photosensitizer to the system. So most of the dyes are not soluble, and they cannot be delivered to the system. So we want to explore carbon nanotubes, multi-walled carbon nanotubes, or we want to explore the Mesoporous silica for the delivery agent as well as for the intrinsic antimicrobial property. So the nanoparticle physical and chemical properties like the small size, high surface to volume ratio allows this technology to come across the barriers to give a better delivery of the photosensitizer. Nanoparticles have been recently used in APDT to enhance the effectiveness of the treatment. So what are the advantages of nanoparticles and photosensitizer together? Large concentrations of photosensitizers for the production of lethal ROS can be done using a combination of nanoparticle. If the photosensitizer is not soluble in water, the encapsulation technology makes them to be soluble and slow release. Supporting the photosensitizer for dimerization and trimerization, it will stop them so that the free active state of the photosensitizer is available, and it reduces the efflux pump. Efflux pump is a mechanism in bacteria. If you use any drug, it will be sent out of the cell. So the nanotechnology will make slow release and sustainable release that makes the bacteria not to activate their efflux pump actively. And selectivity of treatment, localized delivery agents, like we want to target to the wound or to the nervous system or to the lungs or any particular target area, target organ can be done using a nanotechnology platform. In the present study, as I said earlier, I would like to discuss about multi-walled carbon nanotube and the photosensitizer combination in order to kill the bacteria. So you know that carbon nanotubes are emerging as alternative scaffolds for immobilization of antimicrobial agents, either antibiotics, phytochemicals, etc. So carbon nanotubes are long, hollow cylindrical nanostructures wrapped up by graphene sheets. Okay, if you take a graphene sheet, okay, it's a sheet like if you wrap up, it will become a carbon nanotube. If the number of these wraps are increasing, it will become into a multi-walled carbon nanotubes. So CNTs have a unique potential application in various fields because of their unique physical, electrical, and mechanical properties. So such as high aspect ratio and large surface area makes them to be very good property for the biomedical application. So structurally, multi-walled carbon nanotubes consist of multiple layers of graphite superimposed and rolled in one on themselves to form a tubular shape. This makes a promising potential for delivery of drugs, peptides, and nucleic acids for various applications. CNTs, antimicrobial activity. Okay, normally they act on cell membrane, oxidation of cellular components, secondary oxidation of cellular lipids makes them to be antimicrobial in nature. So we want to see their inherent property of antimicrobial property along with the photosensitizer with a light, induced light to see the antimicrobial photodynamic therapy. Single wall nanotubes have been demonstrated to exhibit antimicrobial activity stronger than multi-wall carbon nanotubes. So, but coming to the carrier capacity, multi-wall carbon nanotubes can carry more load compared to that of the single wall carbon nanotubes. So here we taken in this study 2 dyes, that is a toluidine blue and malachite green. So toluidine blue is an cationic phenolphthalein dye, and it shows an absorption band of 630 nanometers. It has a photodestruction activity towards both gram-positive and gram-negative bacterial cells. Whereas malachite green is a cationic dye, and it shows an absorption band at 617 nanometers, and antimicrobial photoinactivation ability of malachite green was against gram-negative and gram-positive bacteria. So toluidine blue and malachite green is already reported for the photodynamic therapy, but the combination of these 2 dyes along with an multi-wall carbon nanotubes was first reported by our group and published in various journals. Considering this, we formulated some objectives. The objectives of this study are synthesis or functionalization of carbon nanotubes, characterization of synthesized carbon nanotubes, conjugation of photosynthesizer with carbon nanotube, and then characterization of photosensitizer carbon nanotube together, that conjugate, and to study the uptake and release of this photosensitizer to the bacterial system, photodynamic inactivation of planktonic cells. Planktonic cells are free-floating cells of bacteria. And then we would like to study the biofilm inhibition also using this composite, photosensitizer and CNT composite. Study the mechanism of action, how this photosensitizer photodynamic therapy is working, and it's a statistical analysis. So highlights of the work I'm going to explain are an effective and enhanced photodynamic inactivation of Pseudomonas aeruginosa and Staphylococcus aureus was achieved by toluidine blue, malachite green, CNT conjugate, antimicrobial and antibiofilm activity of toluidine blue, malachite green CNT composition and identification of Ross generation. Synthesized conjugate can be used in the treatment of infections related to Pseudomonas aeruginosa and Staphylococcus aureus. So first one, I'm going to explain about toluidine blue doped carbon nanotubes and their application. So here, the parameters what we have taken is, we use a diode laser of 670 nanometers. Peak wavelength is 630 nanometers. And this laser is a continuous mode laser with a radiant power of 125 and with an irradiated area of 0.85. And the number of treatments are one time only we treated. It's not because in one time itself we received almost 99% above inhibition of the bacterial cell. So the results, coming to the results, this is the TBCNT conjugate. So we studied the spectral characteristic spectroscopy of CNT. So here in the picture you can see the black line represents the CNT. Whereas the blue one represents the TBCNT conjugate and the red one is the TB. So in black line, an peak at 280 was observed, whereas in the blue red light, you can see in the red line that is for the TB, you can see an wavelength of 630. But when you see the constant TBCNT, both the peaks were observed. It is the indication that the TBCNT conjugate has been happened. And the same was observed under HRTM to see the morphology of carbon nanotubes. So then to confirm the conjugation process, we studied FTR spectrum. So the first one is the CNT FTR spectrum, second one is a toluidine blue, and the third one is a TBCNT conjugate. So when we studied this, you can see there is an at a wavelength of 3451, 1444 and 1458, we got the spectrum for CNT, whereas the spectrum was observed at 3267, 1613, 1535, 626 for TB. Where for TBCNT, you can see both the functional groups in TBCNT FTR spectrum. This clearly indicates that there is a conjugation happened between the photosensitizer, that is toluidine blue, and CNT carbon nanotubes. So again to see the results, validate the results, we studied the Raman spectroscopy, Raman intensity, where the intensity was observed for CNT for 1312 and 1603. For toluidine blue dye laser at 448, 1032, 1394 and 1623, we observed the peaks. The same kind of combination of peaks can be observed in TBCNT, giving an information that, okay, this toluidine blue has been conjugated with the CNT. So we also studied about the photoluminescence spectrum for the dye as well as CNT. You can see here the photoluminescence excitation and emission spectrum for CNT as well as toluidine blue. So then we studied the conjugate. In conjugate also, same kind of emission and excitation spectrums were observed, indicating that conjugation has been observed. So the results of UV visible spectroscopy, FTIR, Raman spectroscopy, as well as the photo emission spectrum indicates that the conjugation been happened. So then we would like to see how much photosensitizer is loaded onto the carbon nanotubes and what is the entrapment efficacy. So for that, we used the dye loading capacity using a formula of M1 by M2 into 100 and entrapment efficacy by a formula of M1 M3 into 100. So we observed a loading capacity of 12.24 okay, for TB, whereas the entrapment efficacy is 48.99. So with this, the radiant exposure was calculated following the laser application. The radiant exposure is equal to PD into exposure time, that is the power density. How we calculated the power density is equal to PW by A. P is the output of power for the laser, and W is the watts, and A is the irradiation area. With these parameters, we studied our photodynamic therapy against bacteria. So you can see here, this figure clearly indicates the uptake percentage of TB to the bacteria, the release, uptake, and as well as the antimicrobial activity. So first A shows that there is a percentage release. The percentage release increased over the period of time. So if you take 120, 180 and further minutes, there is an release there, slow and sustainable release. At the same time, there is an uptake. There was an uptake for the TB, slowly for the bacteria. And the uptake is very good in both the Pseudomonas aeruginosa and Staphylococcus aureus. You can observe in the graphs of B and C panels. D panel shows antimicrobial photodynamic therapy against the bacteria Pseudomonas and Staphylococcus aureus, a significant reduction in the bacteria load. Okay, that is a log 10 reduction we are indicating. So this was observed with the panel D. So with this, we want to see what is the target organelles this photodynamic therapy or by conjugate is observing. So here we studied about the total reactive oxygen species production. So first panel is about the total reactive oxygen species produced when exposed to the Pseudomonas aeruginosa. Okay, there is an increase in the total oxygen reactive ROS production in the TBCNT conjugate when exposed to the light compared to that of the free dye or CNT alone. The second B image shows the reactive oxygen production in Staphylococcus aureus when we treat with CNT, TB, that is a toluidine blue alone, or in a conjugate of TBCNT with and without light. When we expose it to the light or the laser, there will be more reactive oxygen species production compared to that of the free dye or CNT. So the C and D shows the lipid peroxidation. So lipid peroxidation is one of the parameters how the membrane lipids have been destroyed because of photodynamic therapy. So first panel, the C indicates the lipid peroxidation profile of Pseudomonas aeruginosa. D indicates the lipid peroxidation profile of Staphylococcus aureus. Here, a very good lipid peroxidation has been happened after photodynamic therapy. That means the cells are effectively killed by conjugate compared to that of the free dye or CNT after exposure to the laser. So this is the biofilm inhibitory activity. Biofilms are the major problem nowadays for a persistent infection. So we want to study whether the TBCNT conjugate can able to effectively kill the biofilm. So in A, B, C, all the three figures, you can see there is an very good activity of TBCNT when exposed to the light for the biofilm inhibition, and the percentage inhibition is significant compared to that of the free dye or CNT carbon nanotubes compared to the dye after exposure to the light. So we want to visualize how this photodynamic therapy on the bacteria. So we used confocal laser scanning microscopic analysis. to see the destruction of cells. Here we use a dual staining for live and live and dead cells. So you can see first one is about the Pseudomonas aeruginosa. Second one is second two panels are about Staphylococcus aureus. So Pseudomonas aeruginosa control a green colored growth indicate more amount of bacteria live cells are available. CNT, okay, there is a less number of cells. When TB also less number of cells. But when we conjugated with TB-CNT and H panel that is exposed to the laser, there is a more destruction of cells, more red number of cells, more dead cells are available. Similar way, if you take the Staphylococcus aureus, okay, the first panel is about the control, where profound growth of biofilm was observed with green coloration, and whereas H panel, TB-CNT conjugate and exposed to the laser, more photodestruction was observed. The results clearly indicates that TB-CNT conjugate is an effective therapy against Pseudomonas aeruginosa and Staphylococcus aureus infections. So moving to the next study, okay, a similar study, but the dye is different. That is malachite green doped carbon nanotubes. So here in this study, we've taken the same multi-walled carbon nanotubes, but this time we doped with malachite. So the loading capacity and entrapment efficacy for this malachite green doped carbon nanotubes was observed, MgCNT. Loading capacity was observed as 10.31% and entrapment efficacy of 82.51%. This is better than that of the TB-CNT conjugate. So again, we graph the UV visible spectroscopy. Here UV visible spectroscopy shows, okay, CNT, the peaks for CNT, for the lambda max for malachite green and malachite green and CNT. So the lambda max or maximum absorbance that was observed in CNT and Mg was observed in the conjugate. We studied the 10 micrographs to identify the CNTs. So further to validate the result of UV visible spectroscopy, we studied FTIR. So FTIR was graphed between 500 to 4000 wavelength for CNT alone, Mg and MgCNT. The functional groups that are present in the CNT and Mg was observed in the MgCNT conjugate, indicating that there is a perfect conjugation is happened between the Mg and CNT. Similar way, we studied about photoemission spectra individually for Mg, CNT and MgCNT together. In MgCNT, we observed the same kind of photoemission that was observed in Mg alone and CNT alone. So this is about release of malachite green. So percentage release, you can see with increase in time, the percentage of release also increased. So then the B panel is indication of absorbance or uptake. Uptake was also increased with the time in both the bacterial system, Pseudomonas aeruginosa and Staphylococcus aureus. So after entrapment and characterization of conjugation, we would like to see whether the conjugate MgCNT is having any effect on the bacteria. So this is in planktonic cells when we studied CNT with light, without light, Mg with light and without light, MgCNT with light and without light, that is exposure to the laser or not. You can see the last one, that is MgCNT exposure to the laser, is having more log reduction of cells compared to that of the CNT or Mg alone with or without light. So this is about the reactive oxygen species and percentage of protein leakage and inhibition of biofilm. So the reactive oxygen species is the first A. You can see there is an increase in the reactive oxygen species. Okay, the first one is about control. Okay, and with light and without light exposure to the Mg or CNT alone. Whereas when you take the last two bars, okay, here MgCNT when exposed to the light is having more effect when compared to that of not exposed to the laser or laser. So it indicates that the MgCNT can have a better activity in presence of a laser light. So further, we would like to see if the cell damage is happening, any protein is leaking out of the cell. So protein leakage out of the cell is an indication the cell has been damaged and all the intracellular content is leaked out. So you can see more percentage of protein leakage when we expose the MgCNT to the laser compared to that of the CNT or Mg treated bacterial cells, Pseudomonas and Staphylococcus aureus. Furthermore, we studied lipid peroxidation as of my previous slides. In lipid peroxidation also, a considerable, very good lipid peroxidation was observed when CNT was exposed to the laser and wells are treated, Pseudomonas and Staphylococcus. Similar way, the fourth, the D panel indicates that biofilm inhibition. So when Pseudomonas and Staphylococcus were treated with the CNT, MG and MgCNT and exposed to the laser, we have a more bacterial biofilm inhibition compared to that of individual dye with or without light. So this is about reduction of cell viability. So how many cells are dead? To know the reduction of cell viability, a significant reduction in the cell viability and the rhamnolipid production was observed. Okay, through this graph, you can able to see CNT L minus is light, CNT L plus is exposure to the laser, Mg with light and without light. MgCNT without light and with light means with light means laser. So it indicates that MgCNT given to the Pseudomonas and Staphylococcus aureus exposed to the laser can produce more reactive oxygen species and kills more number of bacterial cells. And you can see a reduction in the bacterial cells and bacterial exopolysaccharide production. That is one of the markers for biofilm production. Biofilm formation was also observed in the experiment. So similar to that of my previous study, we want to visualize how much effect is there when this TBCNT conjugate is exposed to the bacterial cell. So confocal laser scanning microscopic analysis, here A, B, C, D and E, F, G, H panels are there. Okay, this first ABC is for Pseudomonas aureus and the other one is B panel is for Staphylococcus aureus. So in control, A and E, you cannot see much bacterial destruction compared to that of the D and H. D and H are MgCNT conjugate exposed to the Pseudomonas aeruginosa and given with the laser light. So where H you can see more red color cells, more photo destruction was observed. Similar way, if you take the B panel, in B also, the H is an indication of MgCNT exposed to Staphylococcus aureus with a laser and a complete destruction, 99.9% destruction of the bacterial cell was observed. So with this, we concluded the results. The field of nanotechnology is now known to as an integral part of medicine with a wide application in drug discovery, drug delivery, and disease diagnostics. One of the emerging drug delivery agent is a carbon nanotube, recently gained eminence as an attractive scaffold in antimicrobial therapy and as a carrier of the drug. We have summarized the efficacy of photoactivated toluidine blue or malachite green with the CNT conjugate for antimicrobial photodynamic therapy against Pseudomonas aeruginosa and Staphylococcus aureus. The ability of this TBCNT and MgCNT conjugate to differentiate the antimicrobial photoactivation against test bacteria contributed to design efficient and potent antimicrobial agents. The conjugate was successfully employed for the reduction of planktonic cells as well as biofilm formation in the bacteria. In future, these nanoconjugates can be used as a potential alternative for the treatment of localized infections. These are the references we followed to study all the photodynamic therapy. And these are the publications. We published the study in various journals like photodiagnosis and photodynamic therapy, photochemical and photobiological therapy, photodiagnosis and photodynamic therapy, and also some other journals about malachite green or toluidine blue conjugated carbon nanotubes. So further, we have an interest on Mesoporous silica nanoparticles. So in order to study the Mesoporous silica nanoparticles, we want to see how Mesoporous silica nanoparticle and the rose Bengal conjugate can able to kill the Candida albicans. So it's an antifungal application. So in this study, we studied the management of that is Candida albicans because it's clinically relevant. Most of the albicans are observed in immunocompromised patients. So even in the fungi, in multi-drug resistance, extensive drug resistant, and pan-drug resistance was observed. This is a major threat to the patients, hospitalized patients, because increased mortality, morbidity, increased hospital stay, and severe economic loss is because of antimicrobial resistance. So in order to prevent this antimicrobial resistance, we have to look for the alternative. So alternative, as I said, okay, we put the alternative as photodynamic therapy. Not only the plant cells, but the problem is associated with the biofilm formation. Biofilms are microbial communities attached to the surface and encased in an extracellular matrix, which is originated from their own bacteria, own fungi. Biofilms are everywhere and can develop in virtually every natural or man-made surfaces. According to the NIH, it is estimated that 80% of human infections are associated with the biofilm-forming microorganisms. Bacterial biofilms and their role in disease have been studied intensively, and in recent years, there is a considerable amount of information is available on the structure and properties of these fungal biofilms, so that we can have an effective drug to prevent the biofilm formation. Coming to the test pathogen, Candida albicans is an opportunistic fungus producing both superficial and systemic infections, especially in immunocompromised patients. Okay, it is one of the serious threat. The ability of Candida albicans to change from the yeast to fermentous is a crucial process for cause and virulence, and subsequently a variety of virulence factors will be produced, which leads to the tissue damage and death. Candemia and candidiasis have been leading type of nosocomial infection with high morbidity and mortality rate around 20 to 40%. The biofilm are resistant to a range of antifungal agents, including amphotericin B, fluconazole, and all other fungal agents. So you can see here the steps involved in the biofilm formation. The cells will be free, they will be attached to the substratum. After attachment, cell-to-cell signaling will happen. Because of the cell-to-cell signaling, the cells will start proliferate and they will produce extracellular matrix to form a biofilm. So this biofilm is resistant to the antibiotics, environmental stress, etc. So, and the top layer of cells, they will rupture the biofilm and they will disperse, and the new biofilm formation will start in other place. So here we used rose Bengal to study the photodynamic therapy. So the optimal molecular design of an antimicrobial photosynthesizer should have several features. So why we have selected rose Bengal is photosensitizers should be non-toxic in the dark, especially towards mammalian cells. To the host, it should not be toxic. They should have good quantum yield of rocks and have a high molecular absorption coefficient at a wavelength where light penetration of tissue is good. Photosensitizers should show selectivity for microbial cells over host mammalian cells, particularly at a short incubation time. And photosensitizers should have a cationic charge, ideally provided by quaternary nitrogen atoms or basic amino groups. So all these features were observed in rose Bengal. So rose Bengal, okay, with an absorption property of 540 nanometers was selected for further studies. So the nanocarrier here we studied is a Mesoporous silica nanoparticle. Among the nano vehicles used in the nanomedicine, Mesoporous silica nanoparticles are of great interest in targeting antimicrobial photodynamic owing to their biocompatibility, high photosensitizer loading capacity, and the ease of surface functionalization. The unique properties of Mesoporous silica nanoparticle, such as they have a controlled particle size and they have a good porosity, their morphology, and high chemical stability, makes them an attractive scaffold for drug delivery, diagnosis, catalysis, separation, and also for sensing. Rapid internalization of animal and plant cells without causing any cytotoxicity is one of the important features of this surface functionalized Mesoporous silica nanoparticle. The biological application of Mesoporous silica nanoparticle includes imaging and diagnostic agents, specificity, dispersibility, and capacity to load higher amount of drug or sensing agent for tracking the cells. Based on this, we formulated some of the objectives. So like Mesoporous silica nanoparticles were taken and functionalized with the amino group. It is characterized using UV visible, FTIR, and other techniques, and then conjugated with a photosensitizer rose Bengal. The conjugation was observed are confirmed using spectrophotometric methods. And then we studied about in vitro uptake and release rate, inactivation, and the mode of action. So these are the results. You can see here, this is a Fti spectrum for mesoporous silica nanoparticle and mesoporous silica nanoparticle after amino functionalization. So after amino functionalization, you can see the introduction of amino group in mesoporous silica nanoparticle compared to that of the MSN alone. So then we studied the conjugation. In a conjugation, mesoporous silica nanoparticle was conjugated with rose bengal with a ratio of 5:1 using ethanol as a solvent. After conjugation, the color of the compound obtained was a pink colored powder. Furthermore, we subjected this to the UV visible spectroscopy, photoluminescence excitation and emission, and FTR spectroscopy. So here, UV visible spectrum, you can see rose bengal is having an absorption at 540, and MSRB, that is mesoporous silica nanoparticle conjugated with rose bengal, also a peak was observed at 540. This is an indication that conjugation has been happened. And photoluminescence analysis, that is photo excitation and photo emission, was observed for for rose bengal and the conjugate, where they are in accordance with each other. So the FTI analysis, we performed the FTI analysis for mesoporous silica nanoparticle and rose bengal alone, as well as the conjugate. So these are the spectral characters, the wavelengths. So you can see MSN with amino functionalization, 469, 800, 59, 1348, 2843, 2953, 3436 are the important functional groups graphed. Whereas for RB, we observed the functional groups at 534, 1238, 1455, and 3411. So the similar kind of functional groups you can see MSN RB at 467, 801, 1342, and 3632. It indicates that there is a good functionalization or conjugation happened between mesoporous silica nanoparticle and rose bengal dye. Further, we studied about loading capacity and entrapment efficacy. So using different formulas, as I explained in the previous slides. So the loading capacity of amino functionalized mesoporous silica nanoparticle was found to be 11.44%, and entrapment efficacy was observed as 58%. So the entrapment and loading are not only important factors. The main important factor is the release of a dye or drug at the target site. So we observed the release pattern up to 3 hours. There is a slow release was observed from 0th hour to the 3rd hour, and the release was almost reached to the 70%. So at the same time, we want to see the uptake percentage. So how much RB, that is a dye alone, will be absorbed by the bacteria or fungi. At the same time, how much uptake is observed with the mesoporous silica nanoparticle. So the uptake of RB to Candida albicans was 36% at the 2nd hour, whereas functionalized and conjugated MSN RB was given to the Candida albicans. The uptake was, cellular uptake was 53%. There is a 20% increase in the uptake after conjugation with the mesoporous silica nanoparticle. So then we studied about antimicrobial photodynamic therapy. First one is the MSN, mesoporous silica nanoparticle, and second one is RB alone. Then MSN RB, that is a conjugate after exposure to the light and without light. So you can see the green color bars are representing with the dark observation without exposure to the laser. The red bars are with exposure to the laser. MSN RB, after exposure to the laser light, there is a more Candida albicans destruction was observed compared to that of the free dye or nanoparticle. So we used a spot assay to determine how much bacterial growth was completely eradicated. D is dark without laser exposure, and L is a light, that is with the laser exposure. So control, you can see the Candida albicans growth. MSN also you can see the bacterial growth. RB also you can see the growth. But whereas when exposure to the laser, MSN RB is having more effect on Candida albicans growth. So we want to see whether how much ROX is generated, reactive oxygen species, when the Candida albicans is exposed to the MSN RB and MSNRB together, conjugate together in presence and absence of light. So in presence of laser light, when MSNRB is given to the Candida albicans, more amount of ROS is generated compared to that of the dark condition or photosensitizer and nanoparticle alone. Cytoplasmic leakage is one of the important features. If the cell has been damaged, cytoplasmic content come out of the cell. So we study the protein leakage. The protein leakage when the Candida albicans is exposed to the MSNRB conjugate with the laser light or without laser light. So we can see a significant reduction or significant protein leakage was observed when MSNRB was given to the Candida albicans and treated with laser. So we want to see further the DNA damage. So these are the DNAs which are extracted from the Candida albicans that is given the MSN, Mesoporous silica, RB, that is Rose Bengal, and MSNRB treated with light or given in the dark condition. So more DNA damage was observed when MSNRB went to the Candida albicans and treated with the laser light. Lipid peroxidation is also one of the indicator. Okay, in lipid peroxidation indicates that how much cell damage has been happened. So lipid peroxidation was observed when the Candida albicans was treated with MSN, that nanoparticle or Rose Bengal dye, or MSNRB conjugate. In MSNRB conjugate, we can see more lipid peroxidation after giving the laser light compared to that of the dark condition. So further, as I said, biofilm is one of the important feature of a bacteria which will get the resistance towards antibiotics. So we'd like to see whether any effect is there towards the biofilm formation. So when we studied the biofilm formation or biofilm inhibition, so MSNRB and MSNRB conjugate given to the Candida albicans, and when MSNRB is given and exposed to the light, laser light, there is almost 90% of the reduction of biofilm was observed. Cell viability also studied by TTT assay. In TTC assay, we observed that there is a complete reduction of bacteria, complete reduction of Candida albicans cells was observed when MSNRB was given to the fungal system and exposed to the laser light. So this is EPS quantification. EPS is, as I said, is an extracellular matrix that is important for the bacteria to get resistant or to form the biofilm. So Candida albicans, the fungi, was exposed to the MSNRB and MSNRB conjugate. And in MSNRB conjugate, you can see more reduction in the Candida albicans EPS production compared to that of either nanoparticle or free dye, Rose Bengal. Confocal laser scanning microscopic analysis. Okay, A, B, C, D are the panels that are in the dark. E, F, G panels are exposed to the laser light. So A is in control. Okay, B is with that of MSN, and C is with the RB, and D and H are RB, C, RB, MSN, RB conjugate and exposed to the laser. You can see more red color cells in the H indicates that a complete reduction or death of Candida albicans cells and exposed to the MSNRB and laser light. With this, we came to a conclusion that Candida albicans is the most common superficial fungal infection in humans. An effective therapy to kill the cells and reduce their biofilm-associated virulence rate. Fluconazole greatly eliminates the yeast cell, but fails when they form the biofilm. So we need an alternative. The present study emphasizes that the prepared nanocomposite, that is MSNRB, significantly enhances the activity of antimicrobial photodynamic therapy against planktonic cells as well as biofilm of Candida albicans. Antimicrobial activity of MSNRB nanoconjugate is more efficient against Candida albicans due to higher photosensitizer binding efficacy, enhanced ROS generation, leakage, DNA damage, and significant biofilm reduction than that of the free dye. Hence, nanocomposite of present study can be used to eradicate the growth and virulence of Candida albicans significantly. So for this study, we referred all these references, and these are the publications published in various journals related to the Mesoporous silicon nanoparticle, the activity against Candida albicans, E. coli, and Staphylococcus and Pseudomonas organism we published. These are all from my lab group. Yeah, thank you. For all this study, one of my PhD scholar, Dr. Raman and Asha Pantori and Anjuviti, they are helped me to study all these MSN, RB, and TNT and Mesoporous silicon nanoparticle preparation and characterization and biological evaluation. Yeah, thank you.
Thank you so
sir.
much.
Ma, if there is any questions, please. Ma. Question and answer session.
Yes, ma'am.
Yeah, yes, ma'am, please.
Good morning, sir. Sir, we have
good
some
morning.
questions from our participant side. Can I start asking the question, sir?
Yeah, you can. You can. Most welcome.
What is the role of entrapment efficiency while doping dye with carbon nanotubes, sir?
Entrapment efficacy gives how much what is called amount of dye is entrapped into the carbon nanotube. If good amount of entrapment is there, then only we can consider them. If entrapment efficacy is just 5%, 2%, or 1%, okay, so we can't able to consider that kind of composite. Entrapment efficacy is one of the important parameter to study to know how much dye is entrapped. So if the more the entrapment, more the delivery of the drug is there. First we have to consider entrapment efficacy, then followed by the release profile.
Okay, sir. Next question. Other than these two dyes, can any other dyes or studied, sir?
Yeah, most of the EJO dyes were studied well for antimicrobial photodynamic therapy. Okay, but in the lab, we did with malachite green, toluene blue, and rose benga. These are the ones studied and reported from my lab.
Okay, sir. It is possible doping CNT development into drugs?
Yeah, so now photodynamic therapy is available for cancer treatment as well as for the microorganisms. In dental caries or dental caries or oral biofilm, any problems are there, goggling solution and photodynamic therapies are available. They are currently in usage.
Yes, sir. Thank you, sir.
Yeah, okay.
Thank you so much, sir, for making your time for us to learn the best.
Sir, just a minute. Thank you very much, sir, for your nice presentation. As we know, dyes are toxic and all, but doped CNT is used for a biomedical application. It's new for us. Thank you for your new answer. Thank you very much, sir.
Thank you, madam, for giving the opportunity. And even I thank my scholars also who supported me with all this data to publish various articles on photodynamic therapy. Very few people are working in India related to photodynamic therapy. I thank all my scholars who helped me to study this photodynamic therapy and publish almost 10, 12 publications related to the photodynamic therapy in highly treated journals. Even I thank the organizers for providing such a very beautiful platform to disseminate my knowledge related to the photodynamic therapy. Thank you, madam.
Thank you, sir. Thank you, sir. Pavitra Devi?
Yes, ma'am.
Please introduce her. Vote of thanks, ma'am.
Okay, ma'am. Good morning, everyone. I thank our management of SDNB Vaishnava College for Women and our speaker of the day, Dr. Busi Siddhartha, who spared time for us in spite of his busy schedule to give this webinar. It is on application of nanoscale particles with photosensitizers for antimicrobial photodynamic therapy. Thank you for the session. It's really an inspiring piece and it was a great opportunity to part of Dr. R. G. Principal thanks Dr. Yasita Lakshmi ma'am, HOD of PG Chemistry, Dr. Suguna ma'am, for extending their guidance and encouragement in all our works. My thanks due to my beloved staff members, student participants, and volunteers for making this session a grand success. I would like to thank and appreciate the participants who choose to attend the webinar with a great enthusiasm and making this event a grand success. Thank you, sir.
Thank you.
Thank you, sir. Thank you, dear students, for this event great success. Thank you all. Please find out the feedback link in the chat box. Please fill it, then you are getting your certificates soon. Thank you.